Hello everyone, my name is Haruka. I'm an application specialist at Cellink Kyoto in Japan. First, I would like to thank organizers for giving me the opportunity to talk at this symposium, and I also would like to thank you all for watching my presentation today. In this presentation, I will talk about how 3D bioprinting works, what we as Cellink offers, and how 3D bioprinting technologies can be applied to study space biology. So first of all, I'd like to talk about how 3D printing and how 3D bioprinting work. So 3D printing, also known as an additive manufacturing, is a process to create 3D object in a layer-by-layer manner. 3D bioprinting is just like 3D printing, but this bio aspect is added. It is an additive biofabrication process to construct 3D bio-objects such as tissue models and all the models from living cells and biomaterials. There are different ways to do this. These are just a few examples of 3D bioprinting technologies. There's inkjet type of printing to create droplets. There's laser-assisted bioprinting, which uses a laser source to deposit liquid biomaterials onto a target substrate in droplet form. And then there's an extrusion-based bioprinting. In extrusion-based bioprinting, it extrudes biomaterials by applying forces mechanically like a screw system or pneumatically using pressures. In light-based printing, stereolithography type of printing is another way to do 3D bioprinting, and it uses light source to cure the photocurable materials during printing process. Bioprinting systems Cellink offers are extrusion-based and light-based. For extrusion-based, we have BioX and the older version of BioX, Incredible Plus, and our newest model, BioX6. For light-based printing, we have Luminex, which uses digital light processing, DLP, and HolographiX, which is based on holographic printing. This is what 3D bioprinting workflow looks like. So you first design the 3D model, and then you choose or engineer bioink to print with. These are the material that you're printing with. And then you do actual 3D bioprinting. Then you incubate for some time, and then move on to downstream processes like imaging, test, and other kinds of analysis. I'd like to point out that the choice of bioink is very important in 3D bioprinting. Things to consider when choosing or engineering bioinks are model and system that you are interested in, and cell material interactions. Cell viability compatibility with the bioink is also very important. Not only cells can survive in these bioinks, but they can behave in the same way or similar way as they are in the physiological conditions like our bodies. Printability is very important. Printability can be influenced by the material properties such as viscosity. Also, some of the material are sensitive to the temperature change. Printing parameters also affect the printability, such as nozzle size and the pressure you're applying to extrude bioinks, and also printing speed, and so on. Cross-linking method needs to be taken into consideration as well. There are different ways to cross-link the constructs that are being printed. There's ionic cross-linking, there's also light cross-linking, and some of the cross-linking is being done with a temperature change. There's also enzymatic cross-linking. As the world's first bioink company, we actually offer a wide range of bioink products. These are some images of the cells cultured in our bioinks. We have chondrocytes, some organoids, and clustering of motor neurons. We also have fibroblasts and keratinocytes. These cells are all cultured in alginate-based bioinks. These mesenchymal stem cells were cultured in gelatin-based bioink. You can also see the heart cells beating. These cells are again cultured in alginate-based bioink. This is a video of BioX. As you can see, it extrudes bioink out of nozzle by applying pressure. I'd like to introduce some of the research applications using 3D bioprinting technologies. Some researchers use our technology to work on tissue models such as an artificial corneal model and a brain model. Also, one of the recent publications of vascularized skin model. These three are all using Cellink extrusion-based bioprinting technologies. This alveolar model is a publication last year in Science by Professor Jordan Miller from Rice University and his group. He used light-based 3D printing technique to create this model, and our Lumina X bioprinter uses the same technology as the work shown in this publication. Some researchers also study disease models, like lung cancer spheroids are shown here. Also viral infection to study influenza. These are both again using extrusion-based bioprinting. Another interesting application is in the area of soft robotics. In these two examples, thermal-responsive materials were used. These materials undergo thermally driven swelling, creating dynamic structures. This adds additional tunability, extending applications in the fields like hydrogel actuators and soft robotics. Food printing can also be done. Using edible inks, food with unique designs can be printed. In some places, food printing applications in elderly care homes are being considered. As you saw in these examples, 3D bioprinting can be applied to so many different fields. So we have many researchers in so many different fields. Very, very exciting. So if 3D bioprinting can be applied to so many different areas, how about space? Why not bioprint in space? Are there any advantages of applying 3D printing and 3D bioprinting technologies in space? One of the main reasons to implement 3D printing and 3D bioprinting technologies in space is to have a self-sustaining system for planetary exploration for the crew during space flight and missions, and also for colonization. During long stay-like missions, it would be better if they could do the on-site productions of the product. With 3D printing technologies, one can print the needed object, for example, tool, on demand, and on-site production can be performed. Currently, how much we can bring on board is still very limited, and this will solve that issue. But also when we consider medical infrastructure during missions, if some medical treatment need to be performed, it is important to be able to do so, as it may not be possible to return to Earth in a reasonable time, and on-board or on-site treatment will be required. If one can fabricate tissues or organs to treat the crew on board, or if people start living or stay on the other planet for a long duration of time and to treat their health issues, a good medical infrastructure is required. Great applications in regenerative medicine, but also in personalized medicine. If you can fabricate tissues or organs using the cells of the specific individual, the care can be tailored to that person. Also, from research perspective, using bioprinting technologies, one can study biology in off-Earth environment. As we leave the Earth, we are exposed to the things that are different from what we are used to on Earth. We can study how biological systems are influenced by these different factors, such as reduced gravity, increased radiation exposure. If certain behaviors are discovered, this can be used as biosensors, and also how these affect human health. If you'd like to expand the length and frequency of our space missions, it is important for us to learn more about how human bodies are affected by the environmental hardship of space. There are active researches related to biofabrication in space already. Some of the researchers study magnetic levitational bioassembly. In this approach, 3D constructs are created by cells or spheroids self-assembling in a paramagnetic fluid in a gradient of magnetic field. This is a scaffold-free approach. There are also researchers studying bioprinting in space using extrusion-based type of printing that I talked about earlier. The challenges associated with biofabrication in space are different environment and how they affect performance of printing and how they affect the samples. These can be the objectives of the studies, as I mentioned in the previous slide, but also the challenges when performing bioprinting in space. Hardware and all the instrument and equipment have to function properly in different environment as well. Also, space limitation. Spaceship only allows certain size and weight. So there are ongoing bioprinting projects in space research, but what can we learn from these researchers and what are they studying? As we get farther from Earth, our surrounding environment changes. We could take advantage of that and study how cells respond to these changes, their behaviors, especially in microgravity and radiation exposure, like higher radiation levels and different radiation composition from what we see on Earth. Some studies have shown stem cells change their behavior under altered gravity. Their fate changes. Also, when performing 3D bioprinting, the materials, the bioinks, how do they behave in different environment? Their viscosity and mechanical properties like stiffness for printing procedures. And in both cases, biology and material, also time. can be added to study the long-time effect by observing the changes over the time. I'd like to talk about ongoing project. Cellink is collaborating with a team of scientists led by Professor Elena Kozlova at Uppsala University to send bioprinted cells to space. So what is this study about? This study is investigating how gravity affects cellular properties. Previous studies have shown that depending on the cell type, gravity affects cell multiplication and differentiation rates in different ways. So this study is to investigate how cells proliferate and differentiate by comparing 2D and 3D, and also comparing them in Earth gravity and in altered gravity. The Swedish Space Agency successfully launched cells that were bioprinted with Cellink bioinks using Cellink bioprinter on the Master 14 rocket on June 24th, 2019. I hope we'll be able to share the results in the near future. I also would like to share an exciting space biology news. NASA has scheduled the launch of SpaceX Crew 1 mission to November 14th, so during this Space Biology Symposium, Crew Dragon will carry 4 astronauts to International Space Station, and one of the crew, he's actually a Japanese astronaut, Soichi Noguchi. He'll be studying biofabrication during this mission. He'll put together the iPSC-derived hepatoblastoma prepared on Earth and vascular-like structure, and see if hepatoblastoma will assemble onto the vascular structure in a reduced gravity environment. If this bioassembly in a reduced gravity is successful, this may have great applications in regenerative medicine in the future. Today I focused on our bioprinting systems, but we also offer many complementary technologies and instruments. These include a low-volume liquid dispenser, single-cell dispenser. We also have imaging system and a microbial reactor. We develop and offer innovative technologies for complete 3D bioprinting solutions. We have many customers all over the world. These are our customers in academia in APEC. We also do live demos. You can book a demo on our website. We also have been doing webinars. These are some of our upcoming webinars. Please join us. For more information about our technologies and product, please visit our website. Thank you all for listening to this talk. We hope to see you soon.